Welcome to Bio 121 Lab. This is the introduction and the procedures for lab number 1, macromolecules and the positive and negative control. In this lab, you're going to be introduced to, among other things, the control, a basic component of experimental design. When a scientist performs an experiment to determine the cause and effect of some phenomenon, it is important to have a point of comparison in order to make sense of whatever results are obtained. So, if you have an unknown mixture and you want to know if it contains sodium chloride, and you have a test that determines the presence of sodium chloride, you could test that mixture to see if you get a positive result, indicating that sodium chloride is there. However, what if your results look like something other than what you were told is the positive response for the test? Positive and negative controls use something that you already know will give a positive or a negative result, respectively. In our sodium chloride example, a solution with table salt in it would provide a known positive response. It would be a positive control. A solution that you know has no sodium chloride in it, for example, plain tap water, would provide a negative response, a negative control. The positive and negative control can be compared against your results to provide you with confidence in your analysis of how your sample responded to the test. You will be using simple tests that indicate the presence of various macromolecular groups. These are what we call colorimetric tests, because of the response of the agent to the chemical group it reacts with results in a specific change in the color from that of the original solution. First, you will be determining what a positive and negative response is for each test using samples of known macromolecules, one corresponding to each test. After you know what the positive and negative responses are for each, you will next be determining the unknown combinations of macromolecules found in 4 different foods. The materials you will need for this lab include a few pieces of equipment and glassware, the reagents, and the samples. The equipment includes a hot plate and a beaker of water with some boiling chips, test tube tongs, a test tube rack with tubes, toothpicks, a paring knife, a spatula, a pestle and mortar, 1 milliliter and 3 milliliter disposable transfer pipettes, a permanent marker, safety goggles, a squirt bottle of deionized water, and paper towels. The glassware includes a 150 milliliter beaker, 4 125 milliliter Erlenmeyer flasks, and a 10 milliliter graduated cylinder. The reagents you will be using are Benedict's solution for determining the presence of monosaccharides, simple sugars like glucose and fructose. Lugol's potassium iodide for determining the presence of polysaccharides, like starch. Biorat's solution for determining the presence of proteins. And finally, oil red O powder for determining the presence of hydrophobic or non-polar substances, like lipids. The samples for the positive and negative controls are deionized water, corn oil, and 10% solutions of egg albumin as the protein, fructose as our monosaccharide, and potato starch as our polysaccharide. The mixtures of the unknown macromolecules are contained in the following foods: potato chips, refried beans, apple, and pepperoni. To determine what a positive and a negative control for each reagent looks like, you will test each of the known 4 macromolecules and the water with each of the 4 reagents. Begin by labeling 5 test tubes numbered 1 through 5. We will use 1 for water, 2 for monosaccharide, 3 for polysaccharide, 4 for protein, and 5 for lipid. We will always test 1 milliliter of the various samples. Transfer the appropriate amount of each sample to the appropriately labeled test tube. The Benedict's test determines the presence of simple sugars, or monosaccharides. The bright blue solution in the presence of monosaccharides in high heat causes a change that results in the colors ranging from bright red-orange, if there's a lot of sugar, to brown and even green if there's only a small amount of sugar. There's also a precipitate that forms and can make the clear solution become cloudy. To perform the test, add 3 milliliters of Benedict's solution to each sample tube with 1 milliliter of sample. Put on your safety goggles and turn on the hot plate, and bring the beaker of water to a boil. Add all of the test tubes with their sample reagent mixture to the boiling water. Allow them to boil for 3 minutes. After 3 minutes, turn off the hot plate and use the test tube tongs to remove the test tubes to the test tube rack. Observe the results for each tube. Tube number 1, the water, represents your negative control. It should not exhibit any change from before the boiling. This is what a sample would look like if there were no monosaccharides present. Comparing each sample to the water, we see that only test tube number 2 looks different. It's turned a bright orange-red and has become very cloudy due to the formation of a lot of precipitate. This makes sense, as test tube number 2 has fructose in it, which is a simple sugar, and should respond positively to the Benedict's test. Therefore, fructose could be used as our positive control. None of the other samples changed because none of them contained a monosaccharide. The Lugol's iodine test determines the presence of complex carbohydrates or polysaccharides. The golden or coppery brown solution in the presence of polysaccharides turns a very dark blue-black color. This happens instantly at room temperature. Once again, add 1 milliliter of the known macromolecular samples to the appropriately labeled clean tubes. Taking the bottle of Lugol's solution, withdraw a pipette full of the potassium iodide. Add 1 drop of the IKI to each test tube of the known samples. Swirl the test tube to completely mix. Using test tube number 1 with water as your negative control, compare each sample with it to identify any change from the honey-colored solution seen in the negative control. You can see that the only sample that is changed is test tube number 3 with the starch. It's turned an inky blue-black. This is what we would expect since starch is a polysaccharide. Therefore, starch would be a positive control. None of the other samples changed because none of them contained a polysaccharide. The Biuret test determines the presence of proteins. The pale blue solution in the presence of proteins and high pH results in a deep violet purple color. This also happens instantly at room temperature. Once again, fill 5 clean, labeled test tubes with 1 milliliter of the known samples. Next, add 1 milliliter of the Biuret solution to each tube. Swirl each tube to mix. Compare the samples to your negative control to identify a change. You can see that only test tube number 4 has changed by turning a deep violet color. Egg albumin is mostly water and protein, and we would expect a positive result from the Biuret test for this macromolecule. So, egg albumin would be a positive control for the Biuret test. The rest remain unchanged. The oil red O test indicates the presence of hydrophobic or nonpolar substances. The dark blackish-brown powder doesn't dissolve in water and merely floats on the surface. In the presence of a hydrophobic compound, the dye dissolves and the solution becomes bright red. This happens at room temperature and can sometimes require vigorous swirling to get complete dissolution of the dye if a nonpolar solvent is present. To your 5 clean test tubes, you'll add 1 milliliter of the known samples to each of the appropriate tubes. Next, using a toothpick as a scoop, get a small quantity of the oil red O powder on the end of the toothpick. Carefully dump this into the sample, starting with tube number 1. Make sure all of the powder moves down to the liquid and then swirl. Repeat this for the other 4 tubes. Using the negative control, notice that the powder simply floats on the surface of the water. Since water is hydrophilic, we don't expect the dye to dissolve in it. This is the same for tubes 2 through 4. Test tube number 5 with the oil, however, is hydrophobic and dissolves the dye, turning a bright red. Thus, corn oil would represent a positive control for the oil red O test. Now you know what both a positive and a negative response looks like for each of the 4 tests. Using this knowledge, you can now determine the presence of various macromolecules that are parts of mixtures in various foods. To determine the macromolecules present in the various food samples will require making solutions of each food. Preparing solutions from the food samples will require grinding pieces of the food sample with a pestle and mortar and then adding water to solubilize the compounds in the food. We'll begin with the apple. Start by cutting a small piece and cutting that into smaller pieces and putting them into the mortar. With the pestle, grind the apple until it's a pulpy mush. Then label an Erlenmeyer flask and add the mashed apple. To that, add 10 milliliters of water and swirl to mix. To make the potato chip solution, place a few chips in a 150 milliliter beaker and add 10 milliliters of water. Allow it to sit and soak up the water for about 15 minutes. After the chips have absorbed most of the water, transfer them to the cleaned mortar. Mash them with the pestle until they look like wet mashed potatoes. Label another flask and add the mashed potato chips. Add 10 milliliters of water and swirl to mix. Preparing the refried bean solution requires opening the can of beans, then scoop out a small quantity into a labeled flask, add 10 milliliters of water, and mix them thoroughly. Finally, for the pepperoni solution, remove 2 slices of pepperoni and cut them into small pieces into the clean mortar. Mash thoroughly with the mortar and pestle. Then add 10 milliliters of water and continue to mash with the mortar and pestle. Transfer the thick pepperoni soup to a labeled flask. To perform the various tests, we will begin by labeling 16 tubes with both the food sample and the test that is to be performed. We will use A to represent the apple, B, potato chips, C, refried beans, and D, pepperoni. Benedict's test is number 1, iodine test number 2, Biuret test number 3, and oil red O test is number 4. Each tube will have a label with a letter for the food and a number for the test. After allowing the food mixtures to settle and solubilize for about 30 minutes, the liquid will be decanted into clean, labeled flasks and further diluted with 90 milliliters of water. This is important in order to reduce the intensity of the color of the sample liquid so that they don't interfere with the color of the test results. 1 milliliter of each sample solution is transferred to the appropriately labeled tube. Then each of the 4 tests are performed on the correspondingly labeled tubes. All of the number 1 tubes get the Benedict's test, all of the number 2 tubes get the iodine test, number 3 get the Biuret, and number 4 get the oil red O test. Test tubes A through D, number 1, gave the following results for the Benedict's test. A1, apple, looks like this. B1, potato chips, looks like this. C1, refried beans, looks like this. And D1, pepperoni, looks like this. Remember, you're looking for both a color change, which can be orange-red to brown-green, and for the presence of any precipitate. Pay close attention to the difference between B1 potato chips and C1 refried beans for both a color difference and presence of any precipitate. The iodine test gave the following results. A2, apple. B2, potato chips. C2, refried beans. And D2, pepperoni. You're looking for a dark blue-black or blue-gray color. The Biuret test gave the following results. A3, B3, C3, and D3. You're looking for a violet or purple color, and make a careful comparison between B3, C3, and D3 for the presence or absence of a purplish color, comparing one to the other. The oil red O test gave the following results. A4, B4, C4, and D4. You're looking for the dye to dissolve and turn the liquid red or a reddish color. Using the positive and negative control results for the original pure macromolecule tests, and the results of the tests on the food samples, you can now determine which of the 4 macromolecules are present in each food. Don't hesitate to go back and look at the positive and negative controls for making comparisons. The appearance of the extracted food solutions in their flasks before testing may also be helpful to you.